I'm going to give a personal talk here, which is my view on some aspects of how we link laboratory cryobiology to ecology in the field. I put this slide in to say something about me, because I guess my name and certainly my face will probably be unknown to most of the people in the room. As John alluded to, I've spent my entire science career working in the Antarctic. This is one of the more beautiful parts of it. It's a place called Marguerite Bay. And my interest has been on how organisms live at low or very low temperatures. However, in the late 70s, early 80s, I did do some genuine cryobiology with John Morris. We were working on means of cryopreserving unicellular algae at the time. So I do have a little bit of experience of cryobiology. But in trying to devise a talk that links lab cryobiology with low temperature ecology in the field, I'm acutely aware of the fact that there are many people in the audience who could give this talk. They'd probably give it a slightly different emphasis from what you're going to hear from me. But also I'm going to have to touch on subjects in which there are people here who know far more about it than I do. So if I upset you or you think I've done something wrong, come and find me at coffee and tell me where I've gone wrong, because this is going to be quite a personal way of trying to link the two fields together. The rough structure, I want to say something about the environmental context of cold temperatures. I then want to say something briefly about how temperature and physiology are linked. And then I'm going to move into the meat of it, trying to look at links between lab cryobiology and ecology in the field. And I'm going to divide it in a very particular way, as you'll see from here. I want to look at unicells first, and then multicells, and then I'm going to try and tie everything together at the end. A couple of people yesterday made allusion to the fact that we sometimes have to justify what we do to outside people. Now I'm retired, I don't have to justify what I do to anybody. It's really great. However, in the time that I worked in the Antarctic, I did have to justify what I was doing time and again, either to members of the public, which is fair enough because they pay for my salary, or to passing chief executives or people from other areas who would think, well, what are these guys doing? They're living at the far end of the earth, they're working in an obscure place, what relevance is it to the rest of us? Well, I think one of the ways that you can point out is to simply say that most of the world is cold. The mean air temperature on Earth, the figures I've given you here are an average between 1979 and 2012. During that period, obviously, the mean temperature of the Earth has risen steadily. But over that period, the average temperature is 14.4 Celsius. That little figure, of course, hides a huge amount of variation spatially, altitudinally, and seasonally. There are quite a lot of parts of the world that are really quite cold, as I'll show you in a minute. However, the largest habitat on the globe, by far, is the sea. And the sea's deep. Almost all of that water is below 4 Celsius. Much of it's below 2 Celsius. So the largest habitat on the face of the Earth, where most of the biota live, is all below 2 degrees Celsius. For life on Earth, living in the cold is the norm. Places like this are the exception. They attract a lot of attention, they're great to visit. That happens to be, in my view, the second greatest place in the world after Antarctica. It's the Namib Desert, but that's a personal view. Most of the world is cold. And the coldest place on the world, as you'll find if you Google it, is Vostok, which is a Soviet station, now Russian Federation station, built at the pole of inaccessibility. This isn't my picture. I'd nearly got to Vostok once, but the plane was cancelled because of bad weather, so this is someone else's picture. When the Americans built the station at the South Pole, an Amundsen Scott station, the Russians built one at the pole of inaccessibility, which is, as the name suggests, the place that's furthest from the sea in the middle of the Antarctic. And the world record air temperature there was recorded, as it says there, in 1983, and it's minus 89.2. Which those of you who do physics will know is well below the temperature at which carbon dioxide freezes. Carbon dioxide doesn't freeze out of the air there because it's so diluted it doesn't nucleate, but it's cold. And this photograph, courtesy of Steve Warren, is actually the log of that day. which is why it's in Cyrillic on the left. It's a Russian. It's their meteorological summary. The ring shows you minus 89.2. This plot here shows you the seasonal variation in temperature at Vostok, which are the white ones at the bottom, and then 2 points in Alaska, Barrow on the coast and Anchorage in the center. The point of this is simply to make two things clear. The first is that although we talk about these record low temperatures, these areas, at least those in the northern hemisphere, have a seasonal variation in temperature, and there are periods of time when they're really, in relative terms, quite warm. The dotted line there shows you zeros just as a point of reference. Vostok, of course, is 6 months out of phase, being in the southern hemisphere, not the north, and it never comes above about minus 30 at Vostok. The other difference between these two is that there is no living biota native in Vostok, but there is in the northern hemisphere. All the cold areas in the northern hemisphere, as I'll show you in a minute, have life. The reason there is nothing living apart from itinerant Russians at Vostok is as much to do with the fact that it's isolated as it is with the fact that it's cold. If we move to the northern hemisphere, a few records. Record low temperature in Alaska is minus 62, came in 71. The record low in North America is very similar, minus 63, that's in the Yukon. In general, because it's more extensive, Europe and Asia are slightly colder than North America, but it's only slight, minus 67.7 in 1933 in Siberia. Very low temperatures are widespread and a significant physiological challenge for a large number of organisms. It's not quite so obscure as passing chief executives occasionally would like to remark. Cold temperatures are real, they're extensive, and they're quite the norm for life on Earth. I'd like to make a slight diversion now to define, to some extent, what it is I want to talk about. This may seem a little of an odd diversion, but there is a point to it. If we want to characterize life, then we need to pull out some essential features, and that particular group of essential features results from the work of two people, one of whom you've probably heard of and the other one you may not have. This is Erwin Schrödinger, who's a well-known physicist who wrote a classic little book in the 1940s called What is Life? It's never been out of print since he wrote it. And this is someone who's probably less well-known. It's one of the fathers of computer theory and cybernetics, John von Neumann. And putting together things that they have thought about from life, about the nature of life, you can come up with a tripartite classification of what is essential about life. The first is a set of instructions, which in modern terminology we call the software, DNA, or in some viruses, RNA. It's hardware, that's the proteins and membranes of the cell, that's the machinery that does the work in the cell. And lastly, there's a flux of energy. To be alive, you need all three. If we look at a little chart here, you can see some distinctions that we can draw. So as an example from a virus has the software, but it's an obligate parasite of cells. It has no hardware of its own, and it has no energy flux. So on the Schrödinger von Neumann classification, viruses are not alive. Now we come to an area that's of interest to cryobiology. If we take an Artemia spore, that has software, has the DNA, it has hardware. It has all the machinery there, but it has no energy flux. It sits there and doesn't metabolize. It's in the domain of interest of cryobiology, but in its state as a spore, it is, by the classification I've just shown you, not alive. But if you put it in the right environment, it becomes alive. So it's an interesting philosophical point, out with this meeting, but it's an interesting philosophical point demonstrating that what we would regard as live is somewhat context dependent. And at the bottom, I've chosen something which is obviously alive. An elephant could be one of anything. I could use an example. It has software, hardware, and energy flux. It's alive. However, those, whilst necessary, are not sufficient. Because thermodynamics tells us that no living entity can remain viable forever, no matter how much energy you put into it. There is one further thing you have to do, and that you have to replicate. And the origin of replication has to do with perpetuating the signal. There is a huge debate as about what came first in the evolution of life, and a replicating molecule is one of the favorites that it is discussed at the moment. We can leave that aside. The critical thing here is that a living organism reproduces. And this is where we first get a distinction between unicells and multicells. In unicells here, bacteria, archaea, and unicellular eukaryotes, I'll come back to those classifications in a minute, it's essentially going from a mother cell to a daughter cell. In the case of a multicell, it's zygote to zygote. So what that enables us to do is distinguish 3 temperature thresholds for life that have a direct relevance to cryobiology. There's a threshold for completion of the life cycle, a low threshold and an upper threshold. Between those two, an organism is alive, metabolizing, and can complete its life cycle. However, there is also a threshold for metabolism, and there is also a threshold for survival. And I can show you those in a sort of pictorial form here. This is a scale where there's an arbitrary scale, low temperatures on the left, high temperatures on the right. Inside the green box there, the animal is between its upper and lower threshold for completing the life cycle. It's doing all the things that we would normally associate with a normal living organism in a habitat. It's metabolizing, it's reproducing. Outside that, it's quite possible that the organism may have a temperature range over which it cannot reproduce, perhaps for energetic reasons, perhaps a physiological system fails, but it can still metabolize, and that's shown in the white boxes at either end. The threshold for survival has no logical association with either the TL or the TM. It could be at the same temperature at which it can complete its life cycle. It may be way below that. An interesting distinction between the two ends is that at the right-hand end, at high temperatures, organisms always reach their TS. There is always a temperature at which survival stops. If you go to the low end, it is possible that some organisms never see their TS. There are plenty of examples of spores which are viable after being held in liquid nitrogen. At the moment, I believe the limit at the far end from which recovery has been possible is 2K. Many organisms, of course, do reach their 2S, but not all organisms necessarily reach it at the far end. So that's the more sort of philosophical and definition side of things out of the way. I now want to say something about the relationship between temperature and physiology. And the picture there was taken in 2006. It's one of our deep field staging posts where we land aircraft to refuel to send them into deep field. One of the reasons that temperature is so important to organism is that it's a state variable, which means that it influences all of the aspects of the environment and the organism itself. Everything about the environment and everything about an organism is temperature dependent. That dependency might be slight or it might be strong, but it's always there. A list of some of the key features that might be of relevance to organisms is shown here. Density, viscosity of water is temperature dependent. The pH of water is temperature dependent. The solubility of gases in water is temperature dependent. The solubility of other solutes in a marine context, carbonate is particularly attracting attention at the moment, is temperature dependent. The molecular motion of all component molecules is temperature dependent, and so on. That's fairly straightforward. It's known from all physical textbooks. You're probably all aware of it. One subtlety that eludes a lot of investigations into temperature physiology and is relevant to cryobiology as well is that that range of temperature dependencies means that you cannot devise an experiment that is fully controlled for temperature. In other words, it is impossible to devise an experiment in which the only thing that differs between the treatment and the control is temperature. If you vary the temperature, you vary the viscosity, you vary the oxygen solubility, you vary everything. So if you have an experiment where you have a treatment that has a temperature term and a control, it's not just the temperature that varies between those two. And if you see a difference between the control and the experiment, even though you've devised an experiment to test for temperature, it might just be something else that's affecting the outcome. A really nice example of this I can give you, because it concerns a good friend and colleague of mine, was investigating over many years the relationship between animal size and temperature. In many aquatic organisms, as you move to colder and colder temperatures, the maximum size of an organism gets larger. And for a long time, people spent inordinate amounts of time trying to work out how temperature had an effect on organism size. And the answer is that it does it because it affects the solubility of oxygen in seawater and fresh water. And the relationship between animal size and temperature is not between size and temperature, it's between size and oxygen content. And that was the thing that eluded people for a long time. There's a powerful lesson there, because we spend a lot of time doing experimental work where we vary the temperature of an organism. We always have to be aware of the fact that it may well be something else that's dictating the results that we see. With that caveat in mind, I've chosen 2 slides here to say something about the effect of organism physiology and temperature. On the left is a graph showing you the relationship between temperature and muscle power output. It happens to be in an Australian lizard, but it could be in anything. It's a general result, but I've shown you there some data for an Australian lizard. On the right is the data for the power output by mitochondria and temperature. There's an important difference between those two, which I'm going to point out now, which is on the left, the data have been arrived at by taking a batch of lizards and moving them up through temperatures. So they've looked at the muscle power output at low temperature, around 15. Then the same lizards have been used to look at power output at 20, the same lizards again at 25, and so on. In order to tease out what is going on in that, you need to take the muscles out of the lizards and look at the individual fibers. And what you find is that what varies with temperature is not the force that the muscle can exert, it's the speed at which it can contract. And because power output is a rate measure, power varies with temperature. The tetanus strength that the muscle can generate is the same in a warm lizard and a cold lizard. The speed at which it can generate that is different between a warm lizard and a cold lizard, and hence the muscle power output is different. That's actually a general result for contractile proteins in general. However, what you're looking at in that left-hand graph is effectively the impact of thermodynamics on muscle contraction. It's the same lizards in all of those data points. It's the same system that is being looked at in all of those data points. You're looking effectively at the impact of thermodynamics on a system. In the right-hand graph, you're looking at the maximum rate that a mitochondria can generate ATP, state 3 respiration rate. It's the maximum rate at which the mitochondria can generate ATP. But each of those data points there, each of these points here, comes from a different organism that has adapted over evolutionary time to live at a different temperature. So effectively, you can say, if I can sort of cartoon it, the left-hand graph is showing you the effects of thermodynamics on a biological system. This right-hand graph is showing you what evolution has or has not been able to do about those challenges. Each of these is a different organism that's had millions of years to cope with the thermodynamics effect over here. The fact that this line has a gradient and isn't horizontal tells you that evolution has not been able to overcome something fundamental about temperature in the rate at which a mitochondrion can generate ATP. Despite millions of years of evolution, organisms living at 0 have mitochondria that can only generate ATP slowly. Organisms that have evolved to live at the top end of the physiological range have mitochondria that are able to generate ATP very quickly. We don't actually know what the rate-limiting step here is. It's believed to be the rate at which the ATPase can operate, but we don't know. It could be the rate at which you can generate the proton flux that needs it. We don't know, but it's suspected to be the rate at which the ATP molecule can turn over. So one of the things that it's important to note is to distinguish a thermodynamic effect from an evolutionary relationship. And the two shouldn't be necessarily confounded. You can't necessarily do an experiment in the lab, see what temperature does to an organism, and assume that that's also an evolutionary relationship. It doesn't work like that. However, trying to step back, as I said at the beginning, and make some sort of broad brush generalizations. There are a few things I'd like to pull out from over a century of physiological work by a huge number of people. The first sort of very broad generalization that I think needs to be made is that there's a difference between living at low temperature and coping with freezing. What organisms do to enable themselves to operate at low temperature is not necessarily the same thing as they need to do to avoid the problems of freezing. In this particular audience, I think that's probably a truism and it probably didn't need saying. But in many low-temperature ecology meetings, it's worth emphasizing. The two are often confounded. They're quite different physiological challenges, which we'll explore in a minute. And as an example, where I think, looking through the literature, the two are often confounded, is in the question of membrane function. What membranes do to enable them, if I can speak that way, what evolution has done to membranes to enable them to operate at zero isn't necessarily the same thing as what membranes have to do in order to avoid the stresses of freezing. The two are sometimes, I might say often, confounded, but they're logically quite distinct, and we need to keep them distinct in trying to make sure we're looking at the right things. In terms of ecology, I would say there are two key physiological or physical challenges when it comes to the phase change of water. The first, as we all know, is that there's a significant change in volume when water moves from the liquid to the solid phase, as shown by a diagram on the left, which was pulled off the web about 2 days ago. The second is that ice, as it forms, excludes the solutes, and the remaining water increases in osmotic strength. Those two are well known to every member of the audience, not so well known in the ecological context. But I thought it might be interesting to put an ecological photograph in here. This is what's loosely known in the Antarctic trade as a brinicle. What it is, you're looking at the seabed. This photograph was taken at McMurdo Sound in shallow water. This is the seabed here, and these, for those of you who can see, the orange blobs on there are actually starfish. Up here is surface ice. It's formed because the air is very cold and the sea is relatively warm. In this particular time of year, the air temperature is about -20, the sea is -2. The ice forms at the surface, and as the ice forms, it excludes the salt from the seawater. And that salt, of course, that salty water can get very cold because it's exposed to air temperatures of -20. So you end up with very cold brine that filters through, drops out of the bottom, and as it comes through, it then freezes the surrounding seawater because it's coming down at about -5, -7, -10. So you end up with a frozen brinicle that's extending down through connects with the seabed, and the frozen brine, or the cold brine and associated frozen water, is on the seabed there. Anything that comes into contact with it generally freezes and dies. So it's an unusual ecological event, but it's a nice, spectacular picture that shows that this exclusion of solutes from seawater actually does have interesting side effects, but they're not relevant necessarily to what we want to talk about now. I've got two slides now where I'm going to try and pull out a very personal view of what I think cryobiology has to say to low-temperature ecology, which is this slide, and the reverse, which is the slide that follows. So much of this here will be all too familiar to the audience, but I'm going to summarize. This is what I've pulled out from a personal summary. Cell survival in vitro is dependent critically on cooling rate. There's an enormous literature that shows that. Despite what I said about membranes on the previous slide, membrane structure and function is clearly linked to cell survival in vitro. Tied to that are water dynamics, which are critically important in determining the fate of a cooled cell. We know from practical experience that survival of those cells can be enhanced by adding cryoprotectants in the suspending medium. We also know that freezing of the cell interior is generally lethal, and that in general, the formation of intracellular ice requires quite fast cooling rates. Here's the reverse. A lot of cells in the natural world, as against the lab, survive by undercooling. Some multicellular organisms allow or possibly even promote freezing of the extracellular fluids. So those are the two sides there. A lot survive by undercooling. Some organisms actually promote freezing of the extracellular fluids. In multicellular organisms exposed to freezing stress, dehydration is critical to survival. In the field, intracellular freezing is generally lethal. I'm aware of two exceptions, or two organisms where intracellular freezing appears to be survivable, but it's rare. Organism survival is dependent critically on cooling rate, and something I'll explore in a minute, no unicell grows, that is, it completes its life cycle, below -20. There's quite deliberately a strong degree of overlap between those two slides. There are some themes that emerge, and I want to look at one or two of them in a little more detail now. But there is one subtlety that I think I'd like to point out. We've heard reference in, I think at least twice I heard yesterday, people say that one of the things cryobiologists can do is look at the natural world, see what organisms in the natural world do, and take that information and knowledge back to the cryobiology lab. In that context, I think it's worth pointing out that what a cryobiologist is trying to do in the lab and what an organism is trying to do in the field, if I can put it in that language, are quite different. Animals and plants only have to survive low temperatures for a short period of time. They don't have to survive for 30 years in liquid nitrogen. They don't have to survive 100 years until someone revives the tissue bank. They only have to make it through to the next summer period. They have to survive a short period, and they only need to do it just well enough. Natural selection doesn't work to produce perfection. Natural selection's job, if you can put it that way, is simply to make sure that this guy is better than its competitors. You don't have to be perfect. You just have to be better than the guys around you. Organisms surviving winter low temperatures are tackling a quite different challenge from organisms that are being treated by a cryobiologist in the lab. There are bound to be some parallels between the two because the physics of the two are the same. But there are some things that are different, and it's, I think, worth bearing in mind that the aims of the two, loose language, are different. Well, one of the things that is clearly linking the two, it had appeared on both of those selections that I gave you, is cooling rate. When I became interested in that, I went into the literature to find data on cooling rate in the natural environment. And you can find lots of general qualitative statements, but you can find relatively little hard data. So I'm starting to accumulate some. And I've got 3 data sets here. I've got more now, but 3 is enough to make the point here. What I've done with these is to take meteorological data, that's the air temperature measured in a Stevenson screen at about this high with a wet-dry bulb thermometer. And I've taken the difference between the temperature at hour 1 and the temperature at hour 2, and then between hour 2 and hour 3. So what we're looking at is the difference in temperature between 1 hourly points of measurement. And you see a plot there of roughly 9,000 such differences over a long period of time from a friend of mine who operates an amateur weather station close to where I live in Norfolk. Now, obviously, most of the time, the temperature at hour n is pretty similar to the temperature at hour n-1, which, of course, is why the peak's around 0. What we're interested in is the fastest rates at which the environment cools, the fastest cooling rate you can observe in the field. And the data I have here are for locally where I live, Rothera Station in Antarctica where I work, and South Georgia in Antarctica where I used to work. Actually, I used to work at Rothera. This is further back in time when I was at South Georgia. And what you see here is that the fastest rate that is observed is really quite slow. It's less than 0.1 K a minute in UK. It's about 0.15 K a minute at Rothera and about 0.3 K a minute at South Georgia. The fastest rate of air temperature change that has ever been recorded meteorologically anywhere in the world is only 2 K a minute. And that was actually, as you can see there, at South Dakota. It was a firn wind that came in. Funnily enough, the same site also has the fastest temperature rise. So it must be something about that particular place in South Dakota that the temperature changes quite dramatically. These temperature changes happen on a daily basis. So here is a temperature record for a site in Antarctica taken by a colleague of mine, and you see the actual temperature plot here going up and down daily, and the rate of change data at the bottom. The key thing to point out here is that these are rates per K per hour, not per minute. So the numbers look big, but actually they're change per hour. The point of that slide is simply to show you that these rates of change occur regularly, but they're still quite low. But they are nonetheless a challenge here. An organism living here is having to do freeze thaw daily and has rates of change which hit peaks of about 0.13 K every day. Those rates, however, are very slow compared with those we use in the lab. I've plotted the same data here, but I've rescaled it. And what you see is the data scaled over on the left-hand side, and 2 dotted lines, which are the threshold rates for survival in 2 whole organism studies. Rana, a frog, and Upis, an insect. What you see there is that the threshold rates for survival, on the right-hand side of the graph, no frogs and no insects survived at those faster cooling rates. At the left-hand side, they survived 100%. So the threshold critical cooling rates are actually quite fast compared with those that you see naturally in the wild. And the arrow shows typical laboratory cooling rates. It's not an original observation to me. I've gone back and quite easily found a reference saying exactly this in 1978 from Keith Miller, who actually did the UPIS work, that the rates we use in the field, the rates we use in the lab, are an order of magnitude faster than those that are ecologically relevant. But there's also another difference between lab and field. And that's shown here in terms of the results of survival of cell types in relation to cooling rate. The left-hand side shows you the classic cryobiological survival curve, a bell-shaped curve. And as we've known for a very long time, the classic explanation for this is the 2-factor hypothesis that says that one suite of factors is operating to reduce survival at low rates, and another suite of factors is operating to lower survival rates to the right-hand side. And that's an oft-repeated experimental result. It's a classical shape in cryobiology. In whole organisms, the shape of the curve is fundamentally different. It's sigmoid. It's a threshold effect. So what that's telling you is that whatever is going on in the cryobiology lab here to give you mortality of cells at very slow cooling rates is not happening to organisms in the field. I don't know what that difference is, and I'd be intrigued to know if other people do. But it's a fundamental division between what's going on in the field and what's going on in the lab. It's become something of a fad in ecology these days that you can't get a comparative paper published unless you control for phylogeny. It's called the phylogenetic police, PC. However, phylogeny is of fundamental importance in trying to understand what organisms do and why they do it. And one of the great revolutions that came out of genomics was the recognition that all organisms on the planet could be divided into 3 high-level domains. Bacteria, used to be called eubacteria, archaea, and eukaryotes. And this division between the 3 is fundamental and basic. It gets a little obscure, of course, because eukaryotes are actually chimeras between these 2. but that's a separate issue. Bacteria and archaea were traditionally regarded as prokaryotes because they look morphologically very similar, and it's only relatively recently they've been recognized as being as distinct from each other as either of them is from eukaryotes. However, in terms of cryobiology, there's a quite different distinction that this is a personal one that I would draw now. And that there are functional domains there that distinguish unicellular organisms from multicellular organisms. And it cuts across the phylogeny. The difference is what happens to those cells when they're exposed to freezing temperatures. In the case of a unicell, once ice forms in the external environment and solutes are excluded, osmotic strength changes, it impinges directly on the cell membrane. Many of these organisms have a cell wall around them, which give them rigidity or some strength and protection, but those cell walls have to be permeable, otherwise the cell can't exchange food and nutrients or waste products with the environment. So although there will be a cell wall or a skeleton in, say, radiolaria, effectively the cell membrane is exposed directly to the changes in the external environment. In the case of a multicellular organism, most of the cells and tissues in the organism are actually exposed to a fluid over which the organism has a degree of control. So the cells and tissues in a multicellular organism are buffered from the changes in the external environment in a way that isn't the case in a unicell. And that unicell could be a bacterium, it could be an archaean, it could be a unicellular eukaryote, as in the picture. This is a functional classification, but it has fundamental consequences, I think, for the way those two groups, domain classifications, those two groups of organisms respond to cryobiological challenges. And I want to illustrate that with some examples about growth and metabolism. I've become recently interested in low temperature limits to growth. And there's a series of examples here. This isn't an exhaustive list, this is simply 4 examples here of lower limits for completion of the life cycle in unicells that you can find in the literature. Arctic sea ice, bacteria have been recorded undergoing cell division down to -15. The lichen, as I guess most of you know, a lichen is actually a symbiosis between an alga and a fungus. In Antarctica has been recorded as photosynthesizing. That's not the same as growing, but photosynthesizing down to -17. A classic food spoiling yeast has been shown to undergo cell division down to -18. And in permafrost, bacterial respiration has been recorded down to -20. If we put all these data together, as was done by Price and Sowers in a PNAS article in 2004, you can see the data plotted here. The upper line here is an Arrhenius plot of the growth data. The line here is a plot for metabolism data, and for the purposes of today, we could ignore the bottom line. What that compilation shows is that there is no data anywhere that indicates microbial growth or metabolism below -20. There are some reports in the literature of metabolism below -20, but they are quite probably down to proxies that are geochemical and solution chemistry at high concentration rather than bacterial respiration. There are no convincing data based on cell number and temperature indicating growth below -20. And that's perhaps not unreasonable given that we've had a century of refrigeration and there aren't any spoilage organisms known that grow below -20. It appears to be a general phenomenon. So it poses the question of what actually is stopping things growing below -20. A suggestion here is that it's vitrification of the cells. As the ice forms, the solutes and cells are excluded from the growing crystal. This is an SEM here. This is where the ice crystals were. What you see in the middle here is a concentration of bacteria that have been excluded from the ice and are in a high concentration solute mixture between the grain boundaries. The cells dehydrate because they're osmotically active and they vitrify. Fernando Fonseca has done a series of measurements of the vitrification temperature of these unicells, and these data are also there in a poster that John Morris has outside, which I recommend you go and look at. The important point to emphasize here is the measurements were done with cells suspended in dilute media and no cryoprotectant, so it's much more relevant to the outside world than it is to cryobiology in the lab. And they show vitrification temperatures which range down, actually 25, minus 25, minus 26. Okay. The advantages of vitrification are that you've got no release of entropy, no change in volume, no danger from gas bubbles, and your metabolism ceases. This is because the viscosity of the internal cell contents is so high that there's no movement of metabolites, there's no movement of oxygen, there's no metabolism. In contrast, if you freeze, there's a strong release of entropy, which you see as latent heat. There's a strong change in volume, which gives mechanical stresses, and the cell usually dies. There's an important distinction here, which I've left this slide in. This is actually in a slide I use in a talk to ecologists, not to cryobiologists, but I've left it in anyway, which is the distinction between bulk vitrification in a liquid, such as shown by that piece of medieval glass on the left-hand side, and colloid vitrification by dehydration, which is what occurs in the cell. And it occurs because the cell is extremely packed inside. It's extremely crowded, and it doesn't take much removal of water before you get close to the vitrification viscosity threshold. One of the interesting things about vitrification as a process in cells is that it links drought and freezing resistance. Ecologists have known for a long time that organisms that are drought resistant are often incidentally also freezing resistant, even though they're living, for example, in highly saline lakes in highly warm places. They will incidentally also be freezing resistant if you take them into a lab. And a functional way of linking those two is vitrification. So can we make any generalizations? Well, I think we can. I think we can say that, firstly, no unicells, that's bacteria, archaea, or unicellular eukaryotes, complete their life cycle below minus 20. I would suggest that as we look more widely, we're going to find that unicellular organisms surviving lower temperatures do so in the vitrified state. As I mentioned earlier, a few reports of apparent microbial metabolism at temperatures below minus 20 probably represent geochemical processes. And a generalization I would make is that the vitrification temperature in these unicells represents a lower limit for life on Earth. An exception, there is always an exception, may well be in clouds, where water droplets can exist in an undercooled state to temperatures below minus 20. Cells can continue to metabolize under those circumstances, and because there's no ice present in the system, they are also not vitrified. But it's a highly unstable environment, and if the droplet freezes, which they often do, then the cells within them die. So lastly, what about multicellular organisms? Well, here life starts to get complicated, which means it also starts to get interesting. In freeze-tolerant organisms, freezing in the extracellular fluids causes dehydration of the cells, well established, which may vitrify through desiccation, or, at very low temperatures, from a low temperature vitrification. Freezing intolerant taxa can continue to metabolize down to very low temperatures, but a recent report suggests that when you reach temperatures of around minus 80, the cells themselves inside these multicellular organisms also can vitrify. It's worth pointing out, however, that there are an awful lot of insects that have critical temperatures which are above zero. This is a compilation of all the data that I know of. It's actually put together by Stephen Chown for the lower critical temperature in insects. It actually also plots the upper critical temperature here, which is not relevant to us today, but the lower critical temperature here for many insects, and you'll see that many insects reach their lower CT and die well before zero. So insects that survive well below zero are only a small subset of insects in general. And it gets complicated, as I say, because some of them, the extracellular fluids freeze, The options for the internal cells there are that they can continue to metabolize, or they dehydrate and vitrify when there's no metabolism, or they live with extracellular fluids under cooling. Cells under those circumstances continue to metabolize them, although they may vitrify at very low temperatures. So there's a whole suite of things here, a whole complexity, which multicellular organisms are capable of that you don't see in the unicells, which is why I made the functional distinction there between the unicellular response to low temperature and the multicellular response to low temperature. The multicellular response is more varied, it's more complex, but it has the advantage that it enables the organisms to live to much lower temperatures than they can do if they were a unicell. One of the advantages of evolving multicellularity is that you've been able to extend the lower limit of life downwards. As an aside, if you look at the upper limit to life, it's dominated entirely by unicellular organisms. Archaea to plus 121, bacteria to plus 93 at the moment. No eukaryote that I'm aware of completes its life cycle above about 65 Celsius. There's probably some out there somewhere, but we don't know of them at the moment. There are, of course, some organisms with particular special adaptations to avoid freezing. And the most famous of these can't be ignored in a talk like this, and that's antifreeze in fish. The particular feature of fish is that their blood is dilute. We don't know why. It has to do with their evolutionary history, but they have a blood which is only about half the strength of seawater. So fish would normally freeze at about minus 0.5. And if you do the experiment with a goldfish, it does freeze at around minus 0.5. And it doesn't matter how many apocryphal stories from your grandmother's pond you read, no fish survives freezing. It's always lethal. No matter what apocryphal stories you can hear about your goldfish ponds in the garden, they're actually surviving anoxia under those circumstances. They're not surviving freezing. However, polar fish can live down to minus 2 because of a complex series of physiological adaptations, one of which is the synthesis of an antifreeze. It's worth emphasizing here that the antifreeze isn't the end of the story. With the antifreeze comes a whole suite of other adaptations, and it's that suite of adaptations that enables the fish to survive below minus 2. But to go back to the point I made earlier on, it's only just. They only have to survive down to minus 2. They don't have to survive minus 20. So they have an antifreeze that works effectively down, gives them a couple of degrees of protection. Insects require much stronger degrees of protection, but I'll leave that to other people to talk about. Antifreeze has actually evolved independently in a number of fish lineages, which suggests that it's not a difficult thing to evolve. But as I mentioned on the previous slide, antifreezes on their own are insufficient. There are adjustments to the excretion physiology, to the circulatory system, there are associated proteins, and it's now become apparent the fish also have to have a mechanism for ridding themselves of ice nuclei. And quite how they do that is a really interesting and as yet unsolved problem. Also, antifreezes, or at least molecules exhibiting thermal hysteresis behavior, have been identified in a large number of organisms, including insects, plant, fungi, and bacteria, though the existence of those in some cases is quite intriguing. Lastly, I want to return to the point about vitrification. I'm going to suggest to you that actually vitrification is going to turn out to be far more widespread in the natural world than we have realized to date. It has been recorded, well known, and going back some time from Arctic plants. But the problem we have is that, of course, in an organism the size of that, actually getting it into the lab to demonstrate vitrification is actually quite difficult. So it's remained a relatively, I think, underreported phenomenon. And I suggest it's going to be something that we find more widespread. So having taken a very personal view through these, it's worth, I think, a last slide to pull together a few pointers to the future. And these are pointers from me, which you will feel well able to disagree with, and in which case I'll see you over a cup of coffee. But I would suggest there's a couple of things that are emerging now. I suspect that vitrification in the natural world will prove to be far more widespread and ecologically relevant than we've recognized to date. Associated with that, I suspect that the role of small molecular weight cryoprotectants, which we've known about for a long time, and proteins with chaperone functions, such as LEAs, something we're learning about much more recently, are likely to become important, or their recognition that they're important in maintaining cell integrity at low temperatures. In other words, what organisms do in the wild to make sure that once they've entered the vitrified state, what they do to ensure that they can return to the fluid state is actually quite complex. It's not simply warming up. And integral to that, I suspect the water dynamics, something which was identified a long time ago, as a number of speakers have already said, and especially membrane physiology, which mediates that water dynamics, are likely to prove to be even more important than we've recognized to date in low temperature survival. Thank you very much. That's a quick survey, and I couldn't have done it without a lot of input from other people, and particularly key people I'd like to acknowledge are on the list there. And also, I'd like to acknowledge the Society for inviting me to give this talk and sponsoring me to come. Thank you very much.